Hello, everyone, and welcome to this webinar organised by the Bayesian section of the Statistical Society of Australia. I'm Matt Wars. I'm a lecturer at Wollongong, and I'd just like to begin by acknowledging the traditional owners of the lands on which we're all meeting. For me, that's the Wadi Wadi people of Dharawal country, and I'd like to pay respects to elders past and present and extend that respect to any Indigenous people who are in the meeting with us today. I'd like to introduce Dr. Alejandra Avalos-Pacheco. She is an assistant professor at TU Vienna in Austria. She did her PhD in the joint Oxford-Warwick Centre for Doctoral Training and a postdoc at Harvard as well. Thanks very much, Ali, and over to you.
Thank you very much, Matt, for the invitation. Thank you, everyone that are here. So today, let me just start sharing my screen. I will talk about the work that I have been recently developed, and it's mainly about integrative factor models and some applications into epidemiologic and cancer genomics. Please feel free to stop me or ask any questions in case you have it. I will try to make this a bit interactive. So the whole motivation of this whole work is that we are interested into discovering these underlying paths from heterogeneous data sets. And why do I mean with heterogeneous? So some motivational studies that impulse or started this work is that we were having data sets coming from, for example, the minerals that and nutrients that people were intaking, and this population was an heterogeneous population coming from people from Latin America. So if we think about it, like we might think that Cubans, Costa Ricans, Mexicans, Central Americans, South Americans, and Dominican Republic, we have things in common, right? Like we like beans, we like corn. So all these nutritional patterns that we will have may have a bit of overlap. But at the same time, it might be that we will have to get also the difference underlying all these nutritional patterns. So the whole idea will be to integrate or stack all these heterogeneous data and try to learn the commonalities or the common dietary patterns that all these populations have in common. And on top of that, also discover the dissimilarities or the dietary patterns that are only unique to those backgrounds, ethnic backgrounds. And the whole goal for that will be to learn how these dietary patterns are related to diseases. And in this case, we will try to look for the links between all these patterns and heart diseases. It's, well, at least we have a Mexican said that it says that we are what we eat. So we are trying to see if we can actually learn something about this, the relationships between diet and health. The second motivation study, it's when we again try to integrate heterogeneous data, but this time coming from populations with cancer. So the whole idea will be to integrate different patients coming from different laboratories where they had been measured their gene expression data. So we have now in this case hundreds of thousands of genes that had been measuring and monitoring. We would like to stack the data that we obtained. It's well known that gene expression has been used to help learning more about the biology of the cancer, and also are a discovery tool to try to see subgroups or subtypes of cancer. So the whole idea will be to stack these studies, do an analysis to that into low dimensional representations because we will have, I don't know, hundreds of thousands of genes that sometimes it's really hard to get to know the feeling what it is. So we would like to get compressed information that in this case will be factors and try to see if we could do a cluster analysis of this data, or if we can try to see of all these genes, try to see the network or structures of clusters of genes and allow to select the most important genes in all these dynamics and see if we can find gene pathways that can help us to understand more the dynamics of the disease. So the first work that I will talk about, it's work that I had developed with David Rosell and Richard Savage, and it's published on Bayesian analysis. So this is an example of the data set that I will be working. As I already said, I'm stacking data, or like putting together data coming from, in this case, they are two laboratories that are recording measurements of patients that have ovarian cancer. And as I already said, we had measured, or they had measured all the gene expression data that they had and obtained that. So the first, let's say, a naive idea would be to just put the data together and then do a low dimensionality reduction technique, in this case, a factor analysis. If we do that without actually any extra treatment, and we plot the first two factors that are the first two, the most important factors in this data set, we can see that we obtain two clear clusters. In here on the left, the stars, and then on the right, the squares. Then doing a deeper analysis to try to understand what are the difference among these two groups, we discovered that the only difference is that they are coming from individuals from different laboratories. So the main driving, or the main source of variation across all these two groups is obviously the difference in technologies, or difference in even technologies and measurements that are doing. If you see here, the two factors are, sorry, the two clusters are different in mean, so they are kind of separated in means, and then also in spreading or in variances. So all these clusters are have two main differences. To this non-biological variation of experimentation, we will call it batch effects. So the batch effects is all these human error, let's call it, or all these technological artifacts that have nothing to do with the biology or the embedded biology that we care about. The problem is that when we don't deal with that, obviously can take us to, in this case, the incorrect conclusion. So I forgot to say something really important. We knew by design, after like taking a deep into the data set, that the patients here in the stars and the squares, we know them to be of the same type of ovarian cancer. So we were hoping them to be exchangeable. So that we were not hoping to see this clear difference there. So, as I already said, these type of batch effects limit our ability to actually see the important biological patterns that we care about. As a first solution here, I will talk about the model that we create, that it's a novel sparse latent factor regression model. The whole idea will be, again, to have the data coming from the different laboratories. Again, importantly, all of them had been measured the same number of genes. Then, after we have this high dimensional data set, we aim to obtain low dimensional factors for all the individuals with hopefully way less dimensionality than the original data. So the original data will be in the order of thousands, And we would like to obtain factors that hopefully are in the order of 10 or something along those lines. Such factors, we would like them that in a model base, we would like to correct these data for systematic biases in mean and variance. So how the model will look like is that we will model the data saying that the data can be explained into the latent structure obtained with the latent factors. This data has been log transformed, so it looks nicely normal. So it can be, it's a realistic assumption to say that we have a normal latent representation of this data set. Then we have the loading matrix that are just going to be the mapping between the low dimension and the high dimensional, that it's a P times Q matrix. And so far, until the padding blue, it's, if you are familiar with factor analysis, that's nothing new. It's just a factor regression model, and then obviously with the error that I will talk a bit later. But on top of that, we put, so we are allowed to measure these differences in mean that I would call the additive batch effects with the parameter beta, that what we observe from the data just a batch indicator of the data saying that individual I to which study it belongs. So it's just a matrix of indicators saying what will be the differences in the means there. So now, kind of for free, we can add in the model other variables that we know that are important, but they don't affect the biology that we have. This is going to be more clear in the nutritional epidemiology example, but in here, let's say that we can get education or the sex that we believe that are not important biologically and can affect a bit the output. So we can correct, let's say, for potential confounders in the latent factors. And we add them just with observed covariates or VI. And we model this effect with the regression coefficients. And finally, so we had correct the differences in mean, we just need to correct differences in variance, right? So we are allowing that each individual, according to the batch that they belong, it will have a different idiosyncratic precision. So we model the errors according to the different batches. So each batch will be allowed to have a different dispersion or variation. So with that, we have a nice model that instead of doing two-step approaches, can in one go correct for batch effects and then give the low dimensional representation of the data. Now we came to another important question, and is that now we have to deal so far with the cardinality. So far I had been saying that we have a low dimensional cardinality, but we haven't said what's the low dimensional cardinality. So as Bayesians, we tend to do this making use of shrinkage or sparse inducing priors. So this is a driven, a really fruitful area of research. And there are several ways to do that via prior distributions with different variances, like the triple gamma prior, the cumulative shrinkage prior, and so on. I will work with a mixture of distribution that I will talk a bit later that are called the spike and slab priors, and we introduce a novel distribution or a novel density that we call it the spike and mom slab, and I will explain this a bit why I mean with that. So the whole idea is that of each of the elements of our loading matrices, we're going to say that each element is, we will add it to the model or not. So it's important or not. That will mean that it's 0 or 1. So it has a value. So if we we add them to the model or not we model this by a mixture of distributions where we said that if these values are important, then we add them to the model with parameter gamma jk, and we model them with a given density. And if they are not important, they will be having really spike this density, and we model them with a given distribution, and then they will have a probability of 1 minus gamma jk of being in that part of the density. So these are called the spike and slab priors, because if we know that they are important, then it will mean that they will be modeling usually with a density with high variance, and if they are, so the density is like slab, it looks slab, and if they are non-important, they are going to be really shrunk to zero, and they are going to model with a spike density. So this is not new. In the literature, this was introduced by Ed George and McLeod when they use a mixture of two normal densities. And with now, if we want to have exact zeros, we can model the spikes and the slabs with Laplace distribution, so they are pointy. And this it has been introduced by Veronica Rakova and Ed George. What we propose is we are modeling the non-zero variables with densities, like in this case, as I already said, slab, that have a high variance. However, when we know that the value is not important, so around zero, they are still putting a non-negligible mass at zero. So we know that around this neighborhood, the probability of that loading to be included should be zero. The problem here is that when we translate this mixture to inclusion probabilities, in the neighbor around zero, the probability of being included is not zero, thanks to this nestedness that we have in these two densities. So what we propose is to model the slab prior with something that is called a non-local density. And a non-local density will look like now, the red lines here, that when we approach to zero, this non-local density will diminish, and then we'll have a zero probability of being included at zero. So what it will allow us is to have two different and well-separated hypotheses and models around zero, that it translates to nice inclusion probabilities, because at zero, the inclusion probability of being, well, the probability of being included, around the neighborhood of zero, it goes to zero. So we are having better separation of the model. With this, these non-local priors were introduced first by Valen Johnson and David Rosell in 2010. And as I already said, we will say that they are absolutely continuous measure with density P theta jk. And it's a non-local prior if, as we approach to zero, then the probability of that factor, of that loading, it will be zero. So how does this non-local prior look like? The good thing about non-local priors is that you can take your favorite density and then just put a penalty at the beginning of the density that it will depend on the moments that we have. So that's why they are called mom, for moments. In this case, we took the decision of working first with normals, but also in the paper that I talked to you about, we also develop Laplace non-local spike and mom slabs. But in this case, we started a slab with the normal, and then we added a penalty at the beginning to make that density goes to zero. So it has now our model the advantages of being sparse. First of all, because it allows us to facilitate interpretation of the factors, just a linear combination of small subset of variables. And we are learning which ones are important without enforcing anything to the model. So with the prior, the data is allowing us, and we are learning which all of those should be 0. So when the sparsity assumption is true, then it can lead to potential gains in estimation accuracy. And in this case, will allow us to work with a really high Q, and then we will learn how many of them are 0. Furthermore, the use of these, because advantages 1 to 3 are true for almost any sparse model. However, on top of that, these non-local priors allow us to have a nice balance between getting to know exactly which are 0 and not going faster to the model to select the true one. And it has been shown that these type of priors have strong frequentist properties for model selection. The problem is that now, if you see here, if we want to do inference on the loadings, then we are adding an extra penalty that is square, and computationally then can be a bit impractical to work with. The solution that we came up is that we developed novel EM algorithms to do inference into those parameters. And after a lot of work, we ended up having good and nice closed form updates that allow us to do via coordinate decent algorithms to efficient inference. And they are packages publicly available at my GitHub, so feel free to open it and destroy that. Thank you. Finally, what we also would like to do, so we have a matrix of loadings. Each of the loadings has an inclusion probability, and we would like to add an extra penalty of that inclusion probability of being added. So in a way that if we are farther away, if we are in column 100 will have a slower probability of being added at column 1 or 2. We will do that via the use of an India buffet process developed by Griffiths and Garamani. So how this will work is that this it's also known for the seek breaking process, but how I see it in the context of factor models is that each of the indicators are drawn or are modeled by a Bernoulli, and each of the Bernoullis will have a different weight for different columns in such a way that each of the columns now will be modeled with parameter beta that depends of the number of column alpha over k. So this translates that if the probability, the inclusion probability, the prior inclusion probability of the first or column indicators, then it will have a uniform distribution to be added. And as we go further away into the dimension, then we will start shrinking more and more towards 0, the probability of inclusion. Finally, we just add gamma prior to the idiosyncratic precision just for conjugacy, as well as a normal. For the same reason, we add a normal prior on the regression parameters. So we start looking at the simulation studies that we obtained. And then in this case, we saw the advantages of just first non-local priors, analyzing data without any batch effects. And we see that when we measure the number of factors obtained, that is the first column here, and we compared our techniques that is in blue with our competitors that are, we compared with a flat prior that is not adding a sparsity, with the normal spike and slab that from the mom's spike and slab and the normal spike and slab, the only difference is the adding of non-local priors there. Then we compare it with a fast BFA that is a spike and slap mixture of priors developed by Veronica Rokova and George, and the classical Lasso BIC when we do factor analysis and then select the number of factors via BIC. So we could see that the cardinality that in this case, if you see for the picture on the left here, the loadings, it's the cardinality is 10. Then we see that non-local priors as well as Lasso are the only ones that can reconstruct properly the latent structure of the data. However, non-local priors, when we measure the covariance reconstruction of each of the factors, it has the smallest covariance reconstruction compared to Lasso, and it has a competitive reconstruction of the actual observed data, and we have a sparser representation of the idiosyncratic errors. This is, sorry, the loading matrices. If we see the second line, we are counting the number of elements that are non-zero, and then we can see that our model was the one that has the smallest zero norm. And on top of that, we are doing this with a really small time, or half of the time that Lasso BIC alternatives. When we did with a higher dimension of the P, of the number of observations, then we have observed the same results. Finally, when we simulate data with arguably the case that we care about, when we have the batch effects, we observe exactly the same things. On top, I forgot to say that in this case, we have one extra competitor that it's what it's done nowadays as state of the art. Then when we have the data coming from different studies, we perform batch effect correction via combat. And after we have done the batch effect correction, then we do low dimensionality reduction. So I would call these two-step approaches here. So when we compare our, in here, true reconstruction, our true versus the reconstruction, we can see that the MOM alternative is the one that is closer to reality. Then the combat or the two-step approaches, it's close to the true and the reconstruction. However, it has a higher variance, so the reconstruction is not that good. And then when we don't do two-step approaches or two-step corrections, then we can see here clear differences between these two studies. So this is just to show that it's a bad idea not to do batch effect correction. Coming to our original data set, when we were talking about ovarian cancer data set, so we applied this method to the data that we were care about, that it's ovarian cancer data sets with 1,000 genes, data coming from 2 different laboratories, and using H as a covariate in the model. This is the first graph that I mentioned when we don't do correction. Then if we do the two-step approaches, then we can still see the differences between the squares and the stars. If you see here, the stars have a higher variance than the squares. Whereas with our method, then there's no difference between squares and stars. They are exchangeable. And that's exactly what we knew by construction of the data that this should look like. But then how do we know that these factors are actually telling something about the biology of the data? So we use these factors to predict survival of the patients. So we compare with the previous methods that I said, the flat, the normal, spike, and slab, the two-step approaches, and the correction with combat and fast Bayesian factor analysis. If you see here, we only observe 4 important factors in comparison to hundreds of other techniques. And with these 4 factors, we obtain a concordance index that this is saying, this is a non-parametric index that it's telling how good the survival prediction is how good is the survival prediction. The higher it is, the better. So we see that with only 4 factors, we obtain concordance in there that other alternatives with 40 or 100 of factors obtained. Furthermore, with only 4 factors, we can get a better, more compact representation of the data than just doing factor analysis that doing the prediction only using one data set or the other. So if you want to learn more about this work, you can scan the paper, or there's extra information of more data sets and more examples in the thesis that I had that I did at work. So I was really happy with the results. However, I was allowing only to vary the studies or I was correcting the batch effects only in means and variances, but I was not letting the studies to differ latent across each one. So we were learning only common factors across all the studies, and we were not allowing the studies to have study-specific factors there. So what we came up, what we would like to now start working is to have now, instead of just common factors, allow each study to have study-specific factors, and here the common ones are green. So now each different study we will allow to have its own study-specific factors. So these type of models have been recently studied, and Chandra just published a paper last year called Subspace Factor Analysis, same as Grabsky, Easy Grabsky, and et al. At some of my colleagues. In this case, I will work with multi-study factor analysis that was developed by Roberta De Vito, Lorenzo Trippa, Giovanni Parmigiani, and because it can be seen as subspace factor analysis is just a special case of multi-study factor analysis. So we would like to work with these models because they are more general. And this how they look like is that now they are modeling each of the individual XIS with the same part that we have here. They are having a common latent structure across model with the loading matrix, the common loading matrix, and each individual will have its own common factor. On top of that, each study will have a latent loading matrix, lambda S, and with the latent factor, and then the error will be exactly the same. What is nice about this model is that it can be easily proved that marginally the covariance structure of each study can be now decomposed into the common covariance part, that is the 5-5 transpose, plus the study-specific covariance part and the idiosyncratic error. So it has been proved that this part in green, the common loadings, are crucial to learn reproducible biological pathways that are different, that are robust and can be mixed with traditional techniques. Like in this case, Roberta De Vito all proved that doing the low dimensionality reduction with different types of cancer, then they could recover these nice genes structure here. So now we're going to work with a different prior, and I will explain why a bit later. In here, instead of having the mixture of the two distributions, we're going to again learn the dimension on the factors, but now with a normal distribution that will have a variance that it's a multiplication of two parts. The first part is the omega jk that I will call the local penalty, that it will be a gamma, and then it will be multiplied by a gamma k, that it will be the global penalty. That now it's a multiplication of gammas according to the column here that we will have. So in a way that as we go further away, we will have more gammas being multiplying to each other. That will mean that we will have smaller variance, and then we will be encouraging the factors to be closer to zero. We do this for the common and story-specific factors. By the way, these multiplicative gamma shrinkage prior has been introduced by Bhattacharya and Donson, and it has been widely used in factor models. Why we use it? Well, I will explain a bit why we use it that. So now again, we have infinitely many factors, and we can have a different degree of a shrinkage depending on the different columns. Traditionally, inference is done by Gibbs sampler. As I already said, they are normal, so they are really convenient because now I can have conjugate posterior, so inference is kind of easy. The problem with that is that doing MCMC implementation of these models can be computationally expensive, especially in the application that we had in mind, that it was in order of 4,000 genes, then this model couldn't run. So are computationally expensive, these methods, and they don't scale well in high dimensions. Plus, they don't take into account these covariate effects that hopefully I convinced you in the other part of the talk that they are important to learn the mean and variances and other extra information. And one last challenge that I had been working that I won't talk in this talk is that unfortunately, though they are really nice and easy to interpret, they have complex identifiability issues that it's another challenge that I have been working with actually a colleague that just moved to Australia to Wollongong, Jack, Jason, David, Rosel, and Roberta. But maybe in the next time that you invite me, I will talk about this later. So coming back to the first challenge. We deal with the MCMC implementation being computationally expensive by developing a new computationally efficient algorithm that builds on machine learning literature. Specifically, we approximate the posterior by a mean field approximation using two algorithms, the CAVI, the coordinate assigned variational inference, and the stochastic variational inference. The whole goal is to have two main points. The first one is that we would like to have a comparable accuracy that the MCMC technique. So we want to be as good as MCMC, but faster, like in a smaller fraction of time or smaller computational cost than MCMC. So this work only for singular factor analysis has been published in a proceedings with Blake Hansen and my colleagues Massimiliano Russo and Roberta De Vito, and it's now has been on archive a version for these single and multi-study settings in those two, CAVI and SBA. So if you are curious, you can feel free and use it. So how, for the ones that don't know how quickly, like a high-level explanation of variational inference, so we care about the posterior of our own non-parameter theta, right? So we would like to find a new distribution that approximates this posterior, that we will call it Q theta given other parameters. And the whole point of this new distribution is that it will have the smallest Kolb-Leiber distance to the posterior that we have here. So we're going to look for this distribution into a family of distributions that are called here Q with cursive letters. And we will start in a given point of all these families of densities, and then we will start moving towards the one that is closest or has the smallest distance via optimization algorithms. So first we have a problem. We need to start looking for a family, which family we will use. So thankfully, thanks to all these conjugacies, and there's a lot of work in this field, we know that we could start working into a mean field variational family. So it will mean that all the parameters, we can use families of distribution that what it considers a partition of the parameters into different blocks. So in a way that each block is independent to each other, and we can just multiply each of the distributions for each block to obtain the distribution that we care about. So it has been, it can be proved, easily proved that then we can approximate the posterior density, making use of this expectation that it will depend on the posterior of the block given everything else and the data. If you're familiar with Gibbs Ampere, it looks a lot like a Gibbs update. So how this will look in practice, well, the dummy example that now we would like to work with this posterior here, and we are now looking these marginals on orange around here of, in this case, Q theta 2 and theta 1. So we would like to obtain these ones. So how it works is that we would like to in here obtain posterior, the posterior in green. We start with a given point in the mean field distribution, family of distribution in blue here. Then we update them, and then we move, closer to the true posterior and closer till we can approximate it. So we iteratively update the posterior parameters and by a coordinate ascent variational methods. So for this, I won't go into details, but just to say that we update each parameter. The problem with this is that some of the parameters, like the ones in red here, will require that will be updated by each observation. And in ultra high dimensional settings, this is not going to be feasible anymore. Even though it's better that MCMC, then it might not be feasible. So the solution will be to now work with stochastic variational inference that it was introduced by Hoffman et al. So the whole idea of the key idea of SBI is that we select a batch size parameter such a way that instead of updating all these observation NS, then we just update a subset or a random selection of these data sets, and at each iteration, we then update the local parameters, or the ones that depend on the number of individuals. And with that, we can approximate the remainder global parameters by a weighted average that we are going to do depending on the step size parameter. So with that, now we propose two novel algorithms. Again, remind you, we want them to be accurate and fast. How do we measure accuracy in this case? Then we have the covariance reconstructor of the data in these simulation exercises, and then the true covariance of the data, this simulation, so we know the ground truth. So we measure the RV coefficient, that is just an extension of our Pearson correlation coefficient, and we want this to be as small as, as closer as possible to MCMC. We measure our method against MCMC. Our methods are the one in blue, SBI, EIC, and ADVI. So we want this correlation to be closer to 1. So the higher the correlation, the closer it is to the ground truth. When we start seeing values of the order of hundreds, then we see that we obtain a good-ish representation, that it's even better than other techniques like approximating the posterior by EM algorithms, or using, that it's the ECM, or using off-the-shelf ADVI algorithms that had been programmed in STAN, that it's this ADVI one. So we are better than other approximation methods, and we are the one that is closest to MCMC. But importantly, in the data that we care about, that it's in the order of thousands here, then we have a good reconstruction, but we can do inference only in 11 minutes. Importantly, we could do inference like in a matter of 2-3 minutes instead of just waiting for half an hour for MCMC. So we call it a win. We were really happy with these results. By the way, my student, Black Hansen, did a lovely work coding all of this in R, so please feel free to use the code there. I will skip this part. We apply this to ovarian gene expression sets. Then we could see that we could run this data set, whereas MCMC couldn't do it in 11 minutes, having the smallest mean square error of the reconstruction and with the best efficacy. In the case, we apply this to try to discover gene expression networks with the sigmas, and then we saw that we could obtain 2 different clusters of the data sets, and then we were really happy because we were, in this case, starting working with 3,600 genes, and we could get the sparsed networks of only 92 nodes that can be divided into 2 groups: the ones that are belonging to the immune system and cell signaling, and then the metastasis and prognosis genes. So we were really happy with these results. Finally, I will quickly work with a model that links these 2 paradigms, the multi-study factor analysis with the factor regression that I mentioned in the first talk. This is work jointly with Roberta De Vito, the one that I will be talking about. And I have been also working this with Jack Houston, David Rosell, and Roberta De Vito. So the whole idea will be to now, on top of that, add covariates to the model that I already know that are not part of the gene expression. So I will skip the simulation scenarios, just suffice to say that we are really good at that. And I will go directly to in the sake of time, to the application. We apply this to nutritional data to try to discover dietary patterns of this heterogeneous population that I mentioned at the beginning. It has been shown that just doing the patterns, dietary patterns per each study don't lead to reproducible nutritional pathways. So we would like to learn then the share and the ethnic specific background in these Latino communities, but also correcting to covariates or confounders that we know that they don't affect the dietary patterns. Like in this case, we could add BMI, gender, education level, alcohol intake, a number of cigarette packs. So with this, we again apply our technique, and we obtain common and study-specific factors, and then we try to see if these factors could help us learn about the overall dietary quality and the probabilities of having a cardiometabolic disease rate by associating each factors with an index that is called AHEI. And this index, it measures the risk of having a disease. The lower the disease of a rate, the lower the chronic disease risk, the higher it is the index. So we would like to have lower indices. So our method was the one that had in the smallest prediction error, which is good. And nicely, we could have 4 different factors that thankfully we could, if we start looking at the proteins or at the different elements that were more expressed, we could name the factors. So the first one, it was for plant foods, the second one animal products, the third one we call it dairy, and the last one seafood. And on top of that, again, we have different study-specific factors. If you see, they look close-ish to each other, but they are some parts that they are not, like in here, the phosphorum in South Americans, or some of them are more expressed than others. To see if these factors were actually telling us something important, then we correlate each of that with different types of food groups. And then it was just a mental check to see that the plant foods were associated with fruits, vegetables, and so on. Animal products were related with meat, meat oil, and so on. The dairy ones with milk, cheese, yogurt. The seafood with fish. And then in here we start seeing that the factors were actually different across these different ethnics or subgroups. And then we see, for example, that the Cubans were related with vegetables, whereas all the other ones were not. And then as well, the Central Americans, the Cuban and Puerto Ricans were correlated with alcohol and snacks. So with that, we use these factors to correlate them with the index. And I was, as proud Mexican, happy to see that the Mexican population were the ones that had a good index. So, sorry, we wanted a high index to have lower diseases, so it goes the other way around. So we wanted high indices. And the Mexicans were the ones that had the highest index. So, yay. Got to Mexicans, at least in this population. So the Latino population living in the US of Mexicans, they were good. And we did a study with the different plant-based animals and food products. So we saw that eating more plant-based increased the index, so it's good. So eating more plant-based foods is good. And it was the other way around with animal products, that eating more animal products decreased the index, so it was bad. So this would be the conclusion. So in this talk, I present to you several techniques to integrate the data set to do two things: obtain low dimensionality factors, and on top of that, correct for batch effects. I show two techniques to do only common factors, or the multi-study factor regression that has common and study-specific factors. I present several ways of learn the cardinality, saying that no local priors were good. And I present several ways of do scalable inference for all this. Important to say that the implementations are here, and hope you can use them. And I show, hopefully, I convince you that these type of models are important to and relevant to in the areas of cancer genomic and nutritional epidemiology. With some extensions, we would like to keep working with sparse priors and start working with new ones, like the base and L1 ball projection of Su and Duan, and other ones like the triple gamma prior, generalized infinite factors, and so on. And on top of that, so far we are having they are common or study specific, that's it. But there's a new work with Izzy Grabsky, Roberta De Vito, Lorenzo Tripan, Giovanni Parmigiani that says, well, maybe there are some parts that are share across study specific factors. So now they allow to have a partially shared factor. So I would like to go and deep into that. And finally, study models that can deal with binary data that are also useful in cancer genomics. I start working with Ising models I learned recently because it's German, with my colleagues at Florence, Andrea Lazzarini, Monia Luparelli, and Francesco Stingo. And I would like to extend this to POTS models. You have experts in Australia like Matt Moores and Katie Mengersen. So I would like to extend this into the multi-study setting. And that's it. Those are the papers. And I finish with time for questions. Thank you so much for the ones that are reached the end.
Thanks very much, Ali. I think there's some questions in the chat, if you can see.
Ah, sorry. Do we need to do any back transformation to interpret the loadings obtained? No, we don't need to do any back transformation. We just take the factors and we can interpret them as they are. So that's a good behavior of this. So it was good. Sorry, I didn't see your question before, Michael. If you have more questions, then I'm open to that.
All right, so just a quick one. So when we interpret the loadings, is the interpretation in reference to the log transform data scale or the original data scale?
Say it again, sorry. My connection broke.
Okay, so the loadings were obtained from the log transform data.
Yes.
So any interpretation we do with the loading, is that with reference to the log transform data or the original data scale?
Well, it will be into the log transform data, but we knew that for construction of this, that the log transfer data will have the same behavior, let's say, in the transform setting with the loadings there. So it will be good enough in this setting. I'm not saying that in every setting, but at least in this setting, it will be good enough to work with these direct loadings there. So it might be different in other settings, but at least in here, we were good to go with that.
Okay, thank you.
Thank you. So Paul, that's a really good question. Have I thought about applying them to single-site data? Yes, the problem with single-site data is that it has a lot of zeros. So then it will be required to do different type of models like the zero-inflated factor analysis or stuff like that. So definitely is on the books. I start working with Roberta De Vito on that, and maybe next year, if I get again invited, or in several years' time, I can start talking about single cell data. Then my slides are available. I don't know. I might need to ask Matt if we can. I'm happy to share them if you send me an email, or if there's like a way that the Australia Society have like a repository, I can try to put them there. So thank you, Tim, for your question. I don't know, Paul and Tim, if I asked your questions correctly. Yeah, I think that's a yes. Welcome.
Great. Well, thanks everyone for coming along for this seminar, and thanks so much, Ali, for presenting it. It was really fantastic.